Hi, I'm Mack Stevens, and I'm an RET teacher here at Norfolk State University. My partner is Melinda, and Nyarko is her last name. We are teachers. I'm in the Advanced Technology Center in Virginia Beach. Melinda is at Granby High School here in Norfolk. And we've been very blessed to be able to work with Dr. Yoon, our advisor. And next slide, please. Our project goals. We broke our project into 3 phases. The first phase, we were trying to get ready for doing the actual research and doing the project. Phase 1 started, we were doing laboratory safety, and we also worked with clean room training. We did phase 2. Phase 2 was getting ready for all the research we were going to do in the project itself. We went to seminars. We learned about all kinds of different projects from people from Morgan State, from Prairie View, and also from Norfolk State. So we had 3 different areas that put in speakers. We learned about webinars. We learned about laboratory skills training and intensive review of background research and articles presentation. Then we went into phase 3, which was the actual research study and also the actual hands-on project that we were working with. Next slide, please. Dr. Yoon actually was in a conference in Korea for the first 3 weeks of our training, so we used that time to prepare. He was in constant contact with his grad students, and they gave us an awful lot of materials to read about to get ready for the project. They also gave us some small projects that we could do, such as soldering, and also we worked with learning what we were going to be doing on the project. Phase 1, the neural imaging laboratory requires a higher level of training than our high school labs, so we had to do a little bit more training to get ready for that, including getting ready for the clean room, which we were able to do a little bit later in the project. Probably week 5 we went into the clean room and we got dressed head to toe, as you can see, just like in this picture, head to toe for the clean room. Next slide, please. Phase 2. This phase of the program provided preparation with webinars, seminars, and lectures. Every Wednesday and Friday we went to seminar. We learned about the projects of other students, and we had to present our own project. We also downloaded programs such as MATLAB, SolidWorks, and also LabVIEW. All 3 of those programs were used extensively in our project, and we had a little bit of experience, but we learned a lot through online tutorials, and also we asked some really good questions to our graduate students who helped us out an awful lot. In the clean room, measuring the channel size of etching silicon chips, we did an awful lot of things, and that was a great experience getting into the clean room. Next slide, please. Phase 3 of our research. The focus of this point in the study is to refine the program frequencies that are detected. So we're trying to figure out which way the person is trying to control the drone through their eye movement and through their brain waves. We actually had this program already made up, so our intense focus was this last part where we were trying to refine it just a little bit. Next slide, please.
Okay. So we picked up on a study that already had the background taken care of. The capturing process involved the bioamplifier system. We used a battery to operate it so that there was no fluctuations in power. We then had the program set up, brain vision recorder for recording the brain waves, and then EEG viewer to transfer that data into a format that MATLAB could read, which changed it into a format that then LabVIEW could read. Next slide, please. So part of our process involved acquiring these brainwave patterns, and to do that, we used a fabric cap and it's designed to hold a ton of electrodes. We only used 8 of them. And the electrodes are put into the cap, and the cap is secured on the subject's head, and it needed to be a bald head, which posed a challenge for us as everybody working in the lab had hair. Nobody was willing to shave their heads. Fortunately, 2 other RETs were bald, and we convinced them that they really needed to participate in our study. So once the cap was placed on, tightly secured, then electrolyte gel was placed into each of these electrodes. As I said, we had 8. We had 4 in the front and 4 across the back to pick up readings from frontal and occipital lobes. All right, once the patterns were established and we had a stable level pattern on the screen, at that point we would actually work with the LED lights flashing, and our test subject would look at the yellow light first, followed by the blue light and the green light. And these were flashing at different frequencies. That is what was being recorded by the Variant Vision recorder, and then that data would be taken and eventually converted so LabVIEW could process it. Next slide. So, captured our patterns. You can see the wave lines there. They're slowly getting more level. Once they're basically parallel, then we can start with the light array, capturing the brainwave pattern, and then, as I said, it has to be processed from here to EEG viewer to MATLAB, and then finally into LabVIEW. Next slide, please. The results of our study, as each of our subjects were looking at the lights, time was recorded for each of the lights flashing, and other than the one trial here, all trials were roughly 90 seconds long. Next slide, please. With these results, when we fed it into LabVIEW, we got positive frequencies on 1 and 4 that were detected. However, our other 2 frequencies did not detect. However, the data, to give you an idea, one of our seminars was on big data, and we found in our data files that there were 114,663 recorded spikes during those 90-second intervals. Can't imagine handling that data by hand. So our current vision of LabVIEW is able to identify the 3 Hertz and the 13 Hertz. It's not able to identify the 5 or the 7. However, we did not get the data processed until July 25th, yesterday, because of the fact that Dr. Yoon had been out of country and there were many, many things he needed to tend to. And so because of that, we really did not have the opportunity to work directly on enhancing this program. Again, one of our biggest problems, finding a bald head. So we know that needs to be a #1 concern for future research. Next slide, please. Okay.
I plan to do two lesson plans from this project that I have developed so far. I plan to do a few more after this, but it'll take a while to work that out. But the first one that I want to talk to you about is how silicon electronic circuit boards are etched. We actually did get to go into the clean room. We actually did get to see the etching process, and so I can develop a number of things from that. We want to show how layers of silicon wafer board, that's going to be the lesson. So we're going to talk about the different layers, and then the steps in etching a circuit board. Hands-on activity, we actually have some circuit board that we can actually put through some acid and etch. It's the traditional method where you actually have to silk screen on it and then put it through acid. But it'll work the same way, and it'll give a really good idea of exactly what they do on a much, much smaller scale. I also want to do a hands-on activity where we are simulating a semiconductor circuit, and that can be found on the PHET website from Colorado. So if you're interested in doing that yourself, you can find that, and there's many, many more models and simulations on the PHET website. The other project that I plan to do, the other lesson I plan to present, is wave scans to MATLAB array. In my class, Modeling and Simulation, we do a lot of bringing in databases and putting them into simulations. And that's exactly what I want to show the students, exactly what we learned through working with this program and exactly how it's done. So those are my 2 lessons. Thank you very much.
Since I'm in the science classroom and not in a technology room, the focus of my lessons, and I have 3 that I will present, has to do with environmental science, which is the science I am teaching. In the first lesson, I want the students to get an overview of what nanotechnology is and how nanotechnology relates into environmental science. Students will do a hands-on search of scavenger hunt, whereby there will be posters around the room that will have a QR code that will identify what area a particular technology is being aimed for. All right, and then we'll, after they've done their scavenger hunt, we'll look at, again, some of these areas where they actually are. Next slide, please. The second part of this lesson will be to get them to actually understand what is a nanometer. How small am I talking when I'm talking about a nanometer? All right, so we have a short video on YouTube which goes over understanding the nanoscale and gives them some common objects like our chart over there with a virus and a red blood cell, so forth, to hopefully have them comprehend a little better. They'll do a think, pair, share. If you're not familiar with that, that's you and your shoulder partner. You think about it individually, the answers to these 2 questions. Then you talk to the person sitting next to you, and you come to some kind of agreement after you have shared your ideas. So 2 things. Can you see a nanometer without technology? And then what would you need in order to see a nanometer? And this lesson would be followed up with a hands-on activity called What is a Nanometer?, whereby students are using rulers and shapes to try to determine the different size of objects. Next slide, please. And the big culminating activity would be a jigsaw, where students would be looking specifically at what nanotechnology can be used to restore damage that we have caused to one of Earth's systems. So our groups, once they have been assigned to a group, will divide back out, with one group looking specifically at the geosphere or the lithosphere, in particular soil restoration. Another group looking at the atmosphere, in particular our greenhouse gases and the whole global warming issue. The third group, the hydrosphere, water pollution or oil spills, something to do with damage that we have caused in that area. And then in group 4, the biosphere, and here it may be something in the medical field, it may be something that we've done to damage some other ecosystem. How do we repair that damage? So this one is just an overview activity. They're not going to get too far in depth on what they're finding, because later in the year, as we concentrate on each of these Earth systems, that's where they will be going more in depth. Next slide, please. So future work, where can this project head? Well, first off, the programming needs to be figured out in LabVIEW. In addition to the fact that we didn't detect all the frequencies, we ran into what I call a single run issue. In other words, each time we wanted to run a data file, we would have to open the program, run only that program, that data file, close the program, reopen. We couldn't load files. sequentially. That takes a lot of time, opening and closing a program. So that definitely needs to be taken care of. And then the next is, how do we enhance it so that we are picking up all of the frequencies that are required? Again, the end product, if we can get that figured out, will be to control that drone sitting up top there. And as Mack said, it could take great strides for helping quadriplegics, anybody with limited movement, if they could control something using just their eye movements. Thank you. Any questions for either one of us?